This is the second recording for Chapter 5. Today we're going to talk about mitosis and cytokinesis. So in the process of the cell life cycle, once a cell has gone through G1, S, and G2 successfully, then it's ready to start the process of dividing the nucleus. In order for the nucleus to divide, we have to have the DNA packaged up in a way that it's protected. DNA by itself is a very tiny, fragile molecule. This double helix would be very easily destroyed if it wasn't properly packaged before a nucleus began to divide. So the DNA double helix will wrap itself around what's called a histone core, 2 times around a core of 8 proteins. These proteins provide a structure and protection for the DNA chain. Now this then begins to twist and coil. This forms chromatin. The chromatin coils then begin to coil in on themselves, making what are called supercoils. And this process of twisting and coiling just continues until all the DNA and its associated proteins is in a structure like this. This is called a chromosome. A chromosome, here we go down one side of the chromosome and the other, and then here is its copy. Remember, in the synthesis phase of the cell life cycle, the DNA copied itself. So this side of the chromosome is an exact copy of that side. Here's another picture of it. We can see that the DNA and the proteins are called chromatin. Here's one side of the chromosome, and then here's the other side of the chromosome. These two sister chromatids are just the same DNA chain, held together at the center by the centromere. So these two sister chromatids, held together right here at the centromere, create one chromosome. When the cell goes through division, it will divide the centromere, splitting the two sister chromatids so that each cell gets an exact copy of the genetic material. Now, there's another structure down here called the telomeres. A telomere is a segment of DNA. It doesn't have genes on it, so it doesn't code for a trait, but it does protect the ends of the chromosome from being eroded away through the process of reading and copying the DNA. The process of mitosis and cytokinesis are going to produce 2 identical daughter cells. We're going to start with one parent cell that has its DNA copied and create 2 smaller identical daughter cells. So after the DNA is divided to opposite sides of the cell, the organelles and the cytoplasm are then going to divide. I've got an animation I want to show you here. Let's do this. So here we have a cell. It's already gone through interphase. It's already copied its DNA. You can see this chromosome has 2 sides, so it's copied. Same with this one and the 2 green ones. And it is at the very beginning of prophase. These are the phases of mitosis in order. Prophase is the first one, so let's see what happens. Notice the nuclear membrane just dissolved away. So now there's nothing containing the chromosomes. And these structures, which we haven't talked about much yet, are beginning to separate. The centrioles, these 2 little guys, are this region contains 2 centrosomes. They're going to separate from one another and go to opposite sides of the cell until they're completely opposite of one another, and they're going to create these little spindle fibers. Let's go to metaphase. Notice they're separating, and the chromosomes line up down the middle of the cell. This represents metaphase, chromosomes down the middle. And the spindle fibers each hook up to that centromere that's in the middle of each of the sister chromatids. Now let's go to anaphase. Spindle fibers shorten, pulling the bundles of chromosomes apart from one another. Now each side of the cell has one full copy of the DNA all to itself. And notice that the cell is beginning to elongate. It's beginning to pull apart just a little bit. Now let's go to the last phase, telophase. Nuclear envelopes reform, and the cell begins to pinch. See right here, the nuclear or the cell membrane is beginning to pinch in. These are protein belts that are going to pull the cell in 2 and is eventually going to divide right down this line. So this cell now has its own nucleus with 1 copy of each of the chromosomes, and this daughter cell has its own nucleus with its own copy of the identical chromosomes. Let's go back over here. Mitosis in plant and animal cells is very similar for much of the process. Cytokinesis, however, has to be different. Animal cells have a soft outer membrane. They're able to pinch in two. But cells in plants have a cell wall that's very rigid and very stiff. It's not possible to simply pinch it in two. So cytokinesis in a plant cell is going to form right here, it's going to form a cell plate. Right down the middle of the cell, this region is beginning to form new cell wall. It happens with vesicles migrating towards the middle of the cell, carrying material that is used to make a cell wall. And as they begin to accumulate and hook together, a cell plate forms, eventually connecting up with the original cell walls on either side, and now we have two completely separate daughter cells. 5-3 regulation we'll get to in the next recording.